Hi everyone, it's time to look at the histology of cartilage and bone, two very specialized types of connective tissue. We're going to start with what we'll call generic cartilage. That's hyaline cartilage, meaning glassy. So we're going to open this up. And the hyaline cartilage is the cartilage that's most frequently found throughout the body. And here we have hyaline cartilage. This is actually going to be the hyaline cartilage at this central strip right here, and we have connective tissue and fat surrounding it elsewhere. So we'll zoom in just a little bit here onto the unlabeled area first, and we can see that we've got adipose cells, these big open spaces here full of adipose tissue that was washed out during the slide preparation process. We have the cartilage here. Hyaline means glassy, and yeah, looks a bit glassy. We have the perichondrium, connective tissue surrounding it, and it has fibroblasts within it. That perichondrium is very important because hyaline cartilage itself is avascular, so the only blood that these cells in the cartilage get will be coming from the blood supply being carried to it inside this perichondrium right here. So those cell layers of fibroblasts and connective tissue, especially type 1 collagen in there, are pretty important. Now if we look at the cartilage itself, we can see that there are cells within here, and they sit within little islands that are called lacunae. Now we might have some chondroblasts, some cells that will become chondrocytes out here on the outer layer that aren't quite yet surrounded by a lacuna, but once they've released enough material into the extracellular space to form this little island or lacuna, they are full-on chondrocytes. So chondroblasts can make it, but chondrocytes have fully become a mature cartilage cell. So we have individual chondrocytes there, but we also have them dividing and forming clusters of cells like this. So we have several daughter cells of a single chondrocyte that divided, and these are called isogenous groups, and they're going to just push away from each other and create new cartilage as they push further away and release their contents into the extracellular space. We can see another group of cells right here that have just divided, and so that's going to be fairly typical of cartilage cells. Now go through your reading and make sure that you can identify the different structures that I've just discussed and which ones are which located here inside the keyed area. We're now going to take a look at a different type of cartilage. This is elastic cartilage, and right here the slide we're looking at was actually drawn from the ear. So if you kind of flip the auricle or the pin of your ear around and notice it kind of wavers back and forth, that's because it is elastic and it's able to actually kind of give and take and bend without breaking. So again, we have the cartilage here at the core, and it looks very much like hyaline cartilage, especially in this hematoxylin and eosin stain. So it's very glassy appearing. We once again have perichondrium and fibroblasts on the outside, on both sides there. We have the cartilage itself here with individual chondrocytes within here. Might see an isogenous group every so often if it's dividing, maybe right there. And the chondroblasts just on the inner lining of that perichondrium. But one thing that is special here is we've got this darker red set of streaks in here. And this is actually staining of elastic fibers, and they're exactly what they sound like. They're fibers that give elasticity to this tissue and allow it to kind of move back and forth. So that's fairly, concise description of it. Hyaline cartilage plus elastic fibers equals elastic cartilage is one way to look at it. We zoom out, we can see some other fun things here. Since this is the ear, we have some skeletal muscle nearby, so remind yourself what skeletal muscle looks like. Peripheral nuclei, striations, etc., etc. And then go in and make sure that you can identify the various keyed structures in conjunction with the handout. Now before we move on, let's take another look at a sample of elastic cartilage, and this is from the epiglottis. Now we can't really just twiddle it back and forth like we did our ear, but it's doing a similar thing. It has to move back and forth to allow us to swallow clearly. So it's going to be located at the back of our oral cavity and kind of helps us exclude food and fluid from our airway. So we look in here. This is a special prep where the elastic fibers themselves have been stained, and they are what's making this very dark purplish black set of structures here. So all that stuff is in the space surrounding these chondrocytes in the isogenous groups. Now, all kinds of cartilage contain type 2 collagen. We generally can't see it very clearly. It's kind of smaller fibers, but that type 2 collagen now is very much associated with these elastic fibers in this specimen and the last one. It's just they're much more visible in this specimen right here. Now, one thing that's interesting about this prep is that we've got cartilage on the outside rim of the epiglottis, but due to aging, we've actually got infiltration of a lot of adipose tissue into that area in between. Sometimes the epiglottis can even ossify if the cartilage becomes bony, but in this case we have quite a bit of fatty infiltration nearby, which you can verify by looking at the large amounts of adipose tissue that are located here too. These are glands, secretory glands, and we're not going to worry about those right now. But here, once again, find the perichondrium, cells in there are the fibroblasts, the cartilage itself, chondrocytes and isogenous groups, and type 2 collagen we can't see, and because it's elastic cartilage, we can definitely see those elastic fibers nice and clear here as these darker purple streaks. We'll now take a look at the third, and for right now, final version of cartilage. This is going to be fibrocartilage, and its name gives you a pretty good idea of what's going on. The slide itself is a little bit of a mess, so let's orient ourselves quickly here. This is the pubic symphysis, where two of the pelvic bones come together on the midline. Because these bones are forming, we have ossification occurring here, more on that in a minute, and we have cartilage right here in the center. So if we zoom in, this is indeed, in blue, hyaline cartilage, the same things we've seen before. If you notice that these cells are all lined up, it's because they're forming growth plates, and again, we'll talk more about that in a second. But all the hallmarks of hyaline cartilage hold true here. We've got chondrocytes and lacunae, type 2 cartilage in between, possible isogenous groups if we see any division happening there, so all the normal stuff. But if we go up here to this darker pink area, this is still cartilage, but something interesting has happened. This is fibrocartilage. The body puts fibrocartilage where it needs to have a lot of resistance to abrasion and wear and tear. So things like our menisci are often made of fibrocartilage. And it's basically hyaline cartilage plus dense irregular connective tissue. So we're going to see fibroblasts. You can see these little streaky cells right here inside the fibrocartilage, and these darker pink streaks that are actually large enough to be visible. Those are type 1 collagen fibers. So these darker pink streaks in here make up that type 1 collagen, which is in assortment with the type 2 collagen that's nearby. Okay, I've never had it go black like that before. Alright, now that we're back, we've got the fibrocartilage present here. We again can see those streaks of type 1 collagen that are there. And one interesting thing is that type 1 collagen takes up a lot of space. It's pretty stout, which means it sometimes pushes these chondrocytes into cords or kind of elongated strips right there. These are different from the growth plates, which we can see over here, where the chondrocytes are very much lined up and proliferating to produce bone from cartilage. And speaking of bone, let's talk about it next. Slide 25 is what's called ground bone. Now in this case, all the living tissue has been removed. The bone was finally ground into a thin strip after being somewhat demineralized, so we can see the organization of compact bone, the bone that's dense and outlines the outermost area of the bones of our body. The main thing that jumps out at you are these various circular or kind of layered structures. Each of these layered structures is known as an osteon, and it's basically the functional architectural unit of compact bone. So let's just pick one that's unlabeled, and yeah, we'll zoom in right here. So at the center of each osteon is a central canal. And sadly, these go by lots of different names, so we'll just use a couple of them. Central canal, also known as an osteocanal or a Haversian canal, and that's where the vessels and nerves that are related to bone are coming from. Bone is living tissue, so it needs to have blood supply and innervation, lymphatic drainage, and so forth. So those canals are carrying blood to the cells of each osteon. But how does it get there? Well, we're going to zoom down here to this already labeled version because it's very nice. The blood gets into these central canals through perforating canals. And of course, these have another name also. They're called Volkmann's canals. But that's how blood is moving from osteon to osteon. We'll zip back up here. And one thing to notice is that we have a central canal, but we also have smaller, fuzzier canaliculi. Those are these dark streaks here. So let's check out what's up with these canaliculi. They're actually open spaces, and they're traveling between adjacent concentric lamellae. Now what that means is we've got layers or lamellae of bone here, and in each layer are several osteocytes. Now osteoblasts lay down bone that then mineralizes, making osteocytes. And here's an individual osteocyte, and these canaliculi are full of cellular cytoplasmic processes of individual osteocytes reaching out to their neighbors. Let's see if we can find a really pretty one. That's not bad. So right here we can see all these canaliculi full of cytoplasmic processes that allow these cells to get nutrients from that central canal, pass it to themselves, pass it to their neighbors, pass it upstream, and so forth. Now because bone is actively remodeled, not only do we have these osteons here that were laid down, we have old osteons that were partially destroyed, filling up some of the space. So right here we've got osteon, 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 and then a little area here that will eventually be remodeled, but it's just a remnant or a fragment of an older osteon that's right there. So that's what we see inside dense bone. Make sure you tour through and identify the labeled structures as well on your own. Alright, now things are about to get interesting. We're going to take a look at how bone forms in the first place. This can usually happen via two distinct routes. The first one's called intramembranous ossification, and that basically implies that we've got bone forming from loose, undifferentiated mesenchyme directly into bone. It doesn't have a cartilage intermediary, so just mesenchyme to bone. This often happens in flat bones, and that's what we're looking at right here. We have membranous bone forming part of the skull, and here this darker pink, kind of almost purplish structure, these are trabeculi or struts of the bone developing here, and this looser cotton candy looking stuff in between, that is the mesenchyme. That's the undifferentiated tissue that is going to become bone eventually. So it's being transferred to bone, and that happens as the osteoblasts located on the outside of these cells, outside of these trabeculae, surround themselves with osteoid, which mineralizes, gives them lacunae, and they become osteocytes. So here are some osteoblasts lined up right here, a little fuzzy, got some others right here. They're going to surround themselves with osteoid. Once surrounded, they'll look like this. They'll have a little lacunae, and they will be a full-on osteocyte, at which point they're going to protect and maintain this little area of bone right in their neighborhood. So we've got those structures present right there inside their lacunae. I should mention we've also got periosteum. Just like we had perichondrium, we have periosteum surrounding bone, and it is dense, irregular connective tissue full of fibroblasts. It brings blood vessels to bone, but as we already saw from the last slide, bone is vascular, so these blood vessels actually go into the bone to supply it directly, as opposed to cartilage, which is avascular and requires diffusion of substances like gases, wastes, nutrients to and from the cells in their blood supply. Alright, now that we've had that little aside, we're going to look here and see if I can find an unlabeled example of our final cell type that we're looking for in this bone, and that is called an osteoclast. I'm seeing several that are labeled right there, F. I want to see if I can find one that isn't labeled so I can point out its specifics to you. An osteoclast is basically a large macrophage-derived multinucleated cell. I'm going to give up and just zoom in on these guys. And these cells, like most macrophage-derived cells, are phagocytic. And in bone, their very specific job is to break down bone so that it can be replaced by new bone. So we have these osteoclasts, multinucleated cells here. What these cells do is fuse themselves to the surface of the bone and then form this what's called resorptive bay, or this little scalloped out area here, where they release a lot of protons, a lot of hydrogen ions, so that we have an acidic environment that erodes the bone, demineralizes it, as well as enzymes like matrix metalloproteases that break down the fibers and other structures here. And these are the cells that break down bone, releasing calcium into the extracellular space and then bloodstream, and allow remodeling to happen. So that's the process of what we have here, this intramembranous ossification. Mesenchyme converts to bone, which then undergoes the process of being resorbed, laid down again, all the way through life. Now that's intramembranous. Let's take a look now at the other variety of bone formation called endochondral. And here we can see a developing digit. So we've got a proximal phalanx here, middle phalanx, and distal phalanx. So these are long bones. And if we take a quick look at this unlabeled portion here, we can see that we've got the head, base, and shaft present right here. So we're going to go up into this area, not only because it's labeled, but it's actually the best example of what's happening in this slide. And it's that we have mesenchyme forming a model of the bone, and that mesenchyme is replaced by cartilage, hyaline cartilage, which is then replaced by bone. Hence the term endochondral within cartilage, ossification. So when this happens, we have a few specific features that are kind of unique. And here, in purple, we've got this very darkly staining hyaline cartilage. So very glassy looking. We can see the individual cells and their lacunae. There's some isogenous groups as well. But growing bone grows because the cartilage proliferates and then eventually ossifies and becomes just part of the bone. We initially have a primary center of ossification in a long bones near the middle of the shaft, right here. It's called the diaphysis. And we'll eventually develop secondary centers of ossification in the epiphyses, or the outer portions. Now that has not happened in this specimen yet, but it would be happening eventually, where we'd get some ossification inside these cartilage areas. So let's return here, and there are 5 basic areas of this growth plate. Yellow, this yellow one here, indicates the zone of reserve cartilage. These cartilage cells, these chondrocytes, are relatively quiescent. They're not really doing much yet, but when they become activated by being closer to the site of growth, we enter this second phase where the red arrow is here, and this is the zone of proliferating chondrocytes. Basically, they are proliferating. The cartilage here is actively growing, got a lot of isogenous groups present here, all in this area. And as they proliferate, they actually start to line up and push their daughter cells in a linear way down towards the ossification centers. And so here, this light blue arrow, number 3, is where we have what's called the zone of hypertrophic cartilage. These lined up cells start to die, and as they do so, they enlarge, and so do their lacunae. So you can see these lacunae are simply much bigger here than they are up here in the reserve zone, and even here in the proliferating zone. Now we get to the fourth part here, this pink arrow. It's a little tough to see, and this part is basically where the chondrocytes have died and their matrix has calcified. So we've got the zone of calcified matrix present right here, and that very quickly enters the zone of resorption, where that calcified cartilage matrix gets chewed up by osteoclasts, sometimes called chondroclasts, but they're basically the same job being done by the same cell type. They're phagocytic cells that chew up this mineralized tissue by releasing acids and enzymes into the area. And this resorptive zone here with the green arrow is where the cartilage is replaced by bone. And at that point, it just becomes part of the shaft of the bone. It's joined the rest of the bone there. It's in contact now roughly with this hollow area in the center, and this area of the diaphysis marks where the primary center of ossification had been, but it's now going to be the marrow cavity. And now we're going to take a quick look at the next slide that's very similar. It's going to be another digit that's forming, but there's a couple things on it that are a little interesting and show up a little better on this next slide. But here it's got some features that are kind of nice that we want to draw a little special attention to. So here's one of these forming long bones. We've got the cartilage capping it on either end. We've got the bone here, primary center of ossification, forming the marrow cavity. Let me zoom in. Feel free to go through the reserve, proliferating, hypertrophic, calcifying, and then resorptive zones. Make sure you can kind of have a fairly decent idea about where those might be located, although some of them are not always very distinct in their borders. But here we can see it's forming that continuation with the trabeculae of bone in here, and we've even got some osteoclasts doing their job and chewing up that matrix and allowing new bone to be laid down. But what I want to focus on in this picture is the area that's outlined in green here. This is outlining where the actual articulation between this bone and this other separate bone is going to happen. So we have this articulation here, and where we have these, we're going to have hyaline cartilage maintain itself. So instead of converting to bone, the hyaline cartilage forming the articular surfaces of these bones will maintain, and it's what's going to allow the bones to glide fairly easily across each other. So lots of hyaline cartilage there, with its type 2 fibers arrayed to diffuse as much of the friction as possible. And speaking of diffusing friction, one specialty of these synovial joints are the tiny little cells that are lining the joint capsule. The joint capsule gets more distinct as we age, but we can see here this connective tissue that's covering the actual joint space. That's the capsule, and its innermost layer is formed by these cells called synoviocytes, and no big surprise, they release synovial fluid, and that helps lubricate the movement of these bones. So synoviocytes present there releasing synovial fluid into this space so that the articular hyaline cartilage of each bone can glide as smoothly as possible across each other. Now, that's where the list of things to investigate ends. I'm going to do one quick view up here of a truly more challenging area. So right here, if you go up to one of these vertebral fetal slides that's forming, we can find a lot of fun stuff in here. So let's just do a quick run through these and just see what tissues we can identify now that we know roughly how bone and cartilage form. So here are 3 adjacent vertebrae that are forming. We have a center of ossification at the core of each of them. The cartilage that made up these bony models is being replaced here by the actual bone. We have growth plates happening as well, but instead of lined up like we have with a long bone, it's basically just forming this circular array out here. So we have the reserve, proliferating, hypertrophic, the calcified matrix, and then resorptive areas all squished into this circular area too. So we're going to challenge just to go try to identify some of those. The nearby vertebrae are separated from each other by fibrocartilage, and we actually have the formation here beginning of these fibrocartilage intervertebral discs. Now they'll have a nucleus pulposus at the center, but right now we can see that we've got the fibrocartilage here. The long streaks of darker type 1 collagen let us know that we're actually dealing with fibrocartilage instead of hyaline cartilage, although the boundary between them is not super abrupt. There's going to be fibroblasts in here as well. Probably these longer spindly guys would be a better ID as a fibroblast. And sometimes people refer to fibrocartilage, cells as having a herringbone pattern, and that's basically just this notched pattern that we see right here. So if you're in the mood for a challenge, go in, try to find all those structures that we just identified on the other slides in this slightly more atypical presentation of a forming vertebral body. All right, good luck to you, and I hope bone and cartilage make a little more sense now than before.